الحالية لمواد التحفيز الضوئي واتجاهها المستقبلي. فليتفضل الدكتور حسام الناصر.
First of all, I would like to thank Professor Dr. Abdulrahman Al-Gazzar for this wonderful introduction. And I would like to thank all organizing committee for this kind invitation to present my lecture entitled Photocatalysis, Future Perspective. First of all, I would like to introduce myself. I am Dr. Hussam Al-Nazir, Professor of Photocatalysis at Photochemistry Department, Chemical Industrial Research Division at National Research Center. Okay, I would like to give an idea about my department, the Photochemistry Department, which is established in 1979 by Professor Dr. Afaf Ali Nada in the framework of DFG project between Egypt and Germany. Professor Afaf Nada was the founder and the first head of and chairman of the Photochemistry Department. The Photochemistry Department research work concerned with photochemistry, photocatalysis, organic synthesis techniques, and their application in industry and environment. My presentation will include the following: introduction on photocatalysis and photocatalyst mechanisms of semiconducting photocatalytic reaction, enhancement of photocatalytic activity, photocatalysis application in environment and industry, national and international collaboration of our research group in Photochemistry Department, and our recent ongoing research project and research team members. And finally, I will conclude all presentation in some statements. So, photocatalysis. Why photocatalysis is the need of the day? Photocatalytic reaction are carried out in green solvent, as well as we use green materials in most of photocatalytic reaction, and we need not to use a large amount of hazardous solvents or temperature or high temperature or pressure. So all photocatalytic reactions are carried out in ambient temperature and room temperature, at ambient pressure and room temperature. So photocatalysis is based on the use of what's called semiconducting photocatalyst that, when irradiated by radiation, is able to perform a redox reaction involving suitable organic and inorganic substrates present in liquid or gas phase. So this figure illustrates the types of photocatalysts used in most of photocatalytic reaction. We see that there are three types. First type is R-type photocatalyst, which promotes only reduction reaction, and O-type photocatalyst, which promotes only oxidation reaction, and there are OR-type photocatalyst, which promotes both oxidation and the reduction reaction. And we see from the figure that titanium dioxide is considered to be the most promising photocatalyst due to its photostability and the chemical stability in both acidic and basic medium, as well as its high photoactivity in most of photocatalytic reaction. So an ideal photocatalyst should be stable, inexpensive, non-toxic, and of course highly photoactive. So this figure illustrates the mechanisms of semiconducting photocatalysts, and we see that every photocatalyst have two energy levels: valence band, which stands for the highest orbital molecular, and homo level, and CB or conduction band stands for conduction band. As we see, when the photocatalyst is irradiated with a certain energy equals to the conduction band equals to the difference between conduction band and the valence band, there is an electron transfer occurs from the valence band to conduction band, resulting in the formation of two active sites where the photooxidation reaction occurs at the valence band active site, and photoreduction reaction occurs as the conduction band of photocatalyst. So this is the all possible reactions that occur on the surface of photocatalyst. For example, titanium dioxide, when irradiated with a suitable energy, photogeneration of electron hole pair occur, and the formation of radicals occur at the surface of photocatalyst, as well as the oxidation of some organics, dissolved organics exists in the solution, could be occur at the hole or valence band of the photocatalyst. So now we are discussing how to enhance the photocatalytic activity. The photocatalytic activity could be enhanced via the following processes: oxygen vacancy formation, doping with inorganic or metal ions, sensitization using diffuse photosensitizer, deposition of metal nanoparticles, and formation of a T-junction between photocatalyst and another semiconducting material or carbon entity like graphene, carbon nanotube, or carbon spheres. So oxygen vacancy formation can be occur by thermal treatment or ultrasonic treatment to the photocatalyst, so resulting in the shift of valence band to a higher energy band. So this will decrease the energy barrier of the photocatalyst. So in this case, the photocatalyst could absorb light with lower energy, like visible light. So as we see in this research article, the researcher here studied the effect of thermal treatment on photocatalytic activity of titanium dioxide, and they found that the thermal treatment at 500 degrees gives the highest absorption power for the photocatalytic reaction of titanium dioxide, and they measure the band gap, and they found that at this thermal treatment, the band gap was very low. Another thing is doping with inorganic species. Also, the doping of photocatalysts like titanium dioxide with copper or nitrogen or sulfur ions could also enhance the photocatalytic activity, since there is also a shift of the valence band to a higher energy level, resulting in the decreasing of energy barrier of the photocatalyst. So the photocatalyst at this moment could absorb energy like visible light with lower energy and higher wavelengths. Also, doping with metal ions also decrease the band gap of the photocatalyst, since the doping with some metals like cerium, zirconium, and iron and copper could also affect the electronic structure of photocatalyst, resulting in also the shift of valence band to a higher level. So this will decrease also the band gap energy of the photocatalyst, and could also absorb energy with or irradiation with lower energy. Also, sensitization using photosensitizer like ruthenium polybipiridine could also enhance the photocatalytic activity of photocatalysts like titanium dioxide. In this composite, the photosensitizer could absorb light with lower energy, like visible energy, and forming electrons which could be injected to the conduction band of titanium dioxide, resulting in the increasing of electrons in the conduction band of titanium dioxide, and so the electron transfer increase and the photocatalytic activity also increase. Also, deposition of metal nanoparticles like platinum, titanium, silver, gold also could enhance the photocatalytic activity of semiconducting material, since the nanoparticles could absorb light with lower energy light, visible light, and then could inject the electrogenerated or electron photogenerated electrons to the conduction band of the photocatalyst, resulting in the increasing of reduction reaction and the conduction band. And also, formation of a T-junction between titanium and another semiconducting also could enhance the photocatalytic activity, since the semiconducting material like cadmium cyanide could absorb light with lower energy and higher wavelengths, and also could be excited and photogeneration, resulting in photogeneration of electrons, which could be injected to the conduction bands of titanium dioxide, and also could increase the rate of photoreduction reaction and formation of radicals. Also, heterojunction formation between titanium and other carbon entity like graphene or carbonate tube or any carbon sphere also could enhance the charge transfer between the titanium, for example, as photocatalyst, and any carbon entity like carbon sphere, where electrons can be transferred from titanium conduction band of titanium to the conduction, the low energy level of carbon entity, and the holes could be injected from the home level of carbon nanotube or carbon sphere to the valence band of titanium. So now we are going to discuss photocatalysis application in environment and industry. So we have a lot of research work concerned with this, with the application of photocatalysis, and we started in 2005 with photocatalytic hydrogen production by water splitting, and this work was with cooperation with Clean Energy Research Institute in Miami University, within Egypt USA project. And in this research work, we succeeded to produce hydrogen from water, from water and dissolved organic compounds, using titanium dioxide in the presence of photosynthesizer like ruthenium bipyridyl, and we have two research articles in this application. Also, we started to study the enhancement of selectivity of photocatalytic reaction via partial photocatalytic oxidation of some sugars like glycerol, and we also succeeded to transfer glycerol to glyceraldehyde without fully degradation of glycerol existed in the solution. And this work was done with the cooperation of photocatalysis group in Palermo University. Also, we studied the photocatalytic oxidation of glucose with simultaneous hydrogen reduction. So, in this research work, we studied both full degradation of glucose with hydrogen production using doped titanium dioxide, and this also work has done with the photocatalysis group in Palermo University. And also the synergetic effect of photo and electrocatalysis has been studied using graphite-supported titanium dioxide, and we studied the degradation of 4-nitrophenol using this photoelectrocatalytic reactor, and we found that the photocatalysis with electrocatalysis also make a higher photocatalytic activity and photodegradation activity. Also, dehalogenation of aryl halides by titanium nanocomposite with parapolyphenylene has been studied for dehalogenation of some halides like bromides and some derivatives of some bromides, and also we found that this nanocomposite has great efficiency in the photocatalytic dehalogenation of these halides without fully degradation. So in this work, we just only transfer halides to non-halides compound and non-halogenated compound. And this work also with cooperation with photocatalysis group at St. Louis University in USA. Now, I will speak about our recent national and international collaboration. We have good cooperation with many national and international research groups. For example, with Professor Vizoroglu, President of International Association of Hydrogen Energy and the head of Clean Energy Research Institute, Miami University, UCA, and the Professor Leonardo Valmusano, the head of Photocatalysis Group in Palermo University, Italy. Professor Andras Dombi, the head of Photocatalysis Group and University of Szeged, Hungary, and Professor Christian Petrier, from University Joseph Fourier, France. Professor Philip Davis, University of Birmingham. Professor Erik Johansson Solom, University of Oslo, Norway. And from Egypt, Professor Sabli Abdel Muttalib, the head of Solar Photochemistry Group in Ain Shams University, and Associate Professor Yasser Atiyah from the National Institute of Laser Enhanced Sciences, Cairo University. Now, we are going to give you an idea about our recent research project. We have this year 3 research projects. The first internal project was funded by National Research Center and entitled Design and Synthesis of Photoactive Hybrids based on nanomaterials as alternative to the important chemicals for water treatment. The second is STDF project entitled Fabrication of Low-Cost Photoactive Antibacterial Hybrids based on local resources for water treatment. And the third international project between Birmingham University and National Research Center in Egypt. And this is Egyptian British project entitled Investigation and Optimization of Design Parameters for Photocatalytic Reverse Osmosis and for water treatment and desalination. Now, I will talk about, and I would like to thank my research project group, Associate Professor Dr. Yasser Mahmoud, the co-PI of our project, Professor Samir Tawfiq Gaballa, Professor Samira Taha Rabia from Photochemistry Department, Associate Professor Ahmad Al-Nazar, and Associate Professor Salman Abdul Salman from Environmental Geology Department, and Dr. Zahra Al-Gohari, Dr. Riham Akram from Photochemistry Department, and Associate Professor Mahmoud Assam and Dr. Ahmed Said from Polymer Department, and Dr. Khulud Hamdi from Water Pollution Department. Now, I would like to conclude all my presentation. Heterogeneous photocatalysis is considered one of the most promising green technology for water treatment, organic synthesis, and hydrogen production. The major advantage of photocatalysis is attributed to the use of mild condition and pressure for different photocatalytic reaction. As I said before, the photocatalytic reaction don't depend on the use of large amount of hazardous organic solvent, but most of photocatalytic reaction are carried out in presence of water or any green materials, and in absence of high temperature and in absence of high pressure. Also, enhancement of photocatalytic activity of photocatalysts could be carried out via oxygen vacancy formation, doping with inorganic species, doping with metal ions, sensitization using suitable photosynthesizer, deposition of noble metals nanoparticles, and formation of heterojunction between titania or another with another semiconductor or titania with carbon entity like graphene or carbon nanotube. Semi-pilot plants based on photocatalysis have been investigated, and we are now designing and implementing a real unit for post desalination and water treatment in our National Research Center. Thank you very much for your attention. Okay.
Thank you, Dr. Hussam al-Nader, for your talk. And now دلوقتي هنتفضل أستاذ Associate Professor Yasser Mahmoud, and his talk is entitled Current Development of Photocatalytic Materials and Their Future Trends. اتفضل يا دكتور.
First of all, I would like to thank the professors and the doctors who are organized this workshop. I will talk about a topic related to photochemistry, which is entitled Current Developments of Photocatalytic Materials and Their Future Trends. My name is Yasser Mahmoud. I am associate professor in photochemistry department, National Research Center. I'm working in photochemistry department since 2003. And now I'm focusing with a good research group in a topic which is called photocatalysis. First, the content of my presentation is about background, short background, and then comparison between different types of catalysis, potential contribution of heterogeneous photonanocatalysis to green chemistry, heterogeneous photonanocatalysts for sense of valuable products, and finally, the conclusion. Here is the background. In 1972, Fukushima and Honda, they recognized the heterogeneous semiconductor photocatalysis. They used titanium dioxide for water splitting and UV radiation. This is the first use of titanium dioxide in photocatalysis. Then, after some years, in 1986, Fuchs and Newton reported the photocatalytic oxidation of some primary amine to imine on titanium dioxide semiconductor. Here is the primary amine, and then they acted, they irradiated this compound by light in presence of titanium dioxide. They obtained and then they obtained the imine product. Therefore, after this work, much effort has been devoted to the application of photonanocatalysts in the sense of organic compounds. Here is in this comparison we would like to present the comparison between different types of concentration and the different type of catalysis, and why the heterogeneous photonanocatalysis is the best, or is the goal of our work. In the first column, traditional homogeneous catalysis, the action condition is drastic condition. It's difficult. The catalyst is difficult to be recovered. The catalyst cannot be used, and consuming large amount of catalyst materials. In the second column, the traditional heterogeneous catalysis, the drastic condition is also required, easily to be recovered, but the large amount of the catalyst is required. The catalyst cannot be reused. In the last column, the third column, the heterogeneous photonanocatalyst, their action could be performed under ambient condition. Could be performed also using solar light or artificial light. So there is no high energy consuming in this reaction. The catalyst can be easily recovered and reused. A small amount of catalyst could be used, and also the water could be used as solvent. Therefore, this catalyst, this type of catalyst, could be a green axis for organic transformation. Here is in this slide, we can show the 12 principles of green chemistry. The scientists agreed about if you are working in green chemistry, they classified the green chemistry in 12 principles. Therefore, in this slide, I would like to show you about how heterogeneous photonanocatalysis could be as a green chemistry tool or a green chemistry technology. We can show about we can look at the heterogeneous photonanocatalysis, the slide at the right hand slide. The low amount of catalyst is loaded in the reaction, which agree with principle number 1 and 9. The catalyst can be easily recovered and reused, principle 1 and 2. Solar light or artificial light used leads to low energy consumption, principle which can agree with principle 6 and 7. High conversion and yields for giving the product, which agree with principle 1 and 8. Water is often used as solvent. Benign substances are used as auxiliaries, which agree with principle 3 and 5 and 12. Degradation of organic pollutants agree with principle 10. Safe oxidation and reduction process agree with principle 3 and 12. Facility to detect the formation of the products and by analysis, which agree with principle 11. Therefore, we can say about that the heterogeneous photonanocatalysis agree with the 12 principles of green chemistry. Here is our recent publication, which is a review article. We tried to write a guide for the scientists or the researchers who are working in heterogeneous photonanocatalyst related to organic transformation. You can find it in Current Organic Sciences journal. This work is written by me, Yasser Mahmoud, and Yasser Atiyah, and Simon Nazar, Eric Solom. Here is the mechanism of the heterogeneous photonanocatalysis. As it was illustrated previously by Professor Simon Nazar in the last presentation, in his presentation, when the photocatalytic material excited by light, the electron could be transferred from valence band to conduction band, and then The electron in the conduction band could be transferred also to the substrates to form products. Therefore, this the process could be make transformation or the action, the organic reaction to obtain products. Also, if you need to process to make organic reaction or to make reaction, you should to look at some points, two important points should be taken in account in your account, which is the efficiency of photonanocatalyst, the selectivity of the reaction for obtaining valuable organic compound as target product. Hence, from this point, numerous strategies for obtaining selective organic transformation were demonstrated that followed by development. This the most research work in this field. They are working in the development of new method for preparation of new organic product, modifying the structure and surface surface characteristics of new or known photocatalysts. This topic in this slide, we are talking about heterogeneous photonanocatalysts for synthesis of valuable products. This in our work. We synthesized the vitamin B3. Vitamin B3 was effectively produced with conversion 100% and yield 90% through photocatalytic oxidation of pyridine 3-methanol on silver zinc oxide graphene. Photocatalyst under visible light irradiation in presence of tertiary butyl dimethyl silyl chloride. In this paper, we made for the first time oxidation from the alcohol, the primer alcohol to the carboxylic acid within 30 minutes by using photocatalyst, by using visible light. And this work was published in Research Chemical Intermediate in 2016. To maximize the value of this work, we made the application of vitamin B3 to mix between the senses and the application. So therefore, we studied the biological activity of vitamin B3 as wound healing agent, and we found that vitamin B3 was good, excellent wound healing agent in comparison with selenium nanoparticle. Therefore, we just made the senses and the application on these products. Therefore, we needed to make valuable product for application. In another research article, we made the senses of paracetamol. Of course, we are know about paracetamol, which is pain relief and is used in as pharmaceutical product. Why we are thinking about paracetamol? That because the previously reported approaches for the sense of paracetamol needs drastic condition or sometimes need long time for production of this compound. The first route it can be produced from paranitrophenol in presence of platinum nanowire and in presence of acetic acid and by heating 100 degrees Celsius and in presence of hydrogen for 24 hours. So it is very drastic condition. And the second route from parahydroxy acetophenol by the reaction with hydroxylamine, and then the second step, which is Beckman arrangement. So you can see it's 2 steps and the high temperature for long time. From hydroquinone, the third route, from hydroquinone, the ammonium acetate to form this intermediate, which is transformed to paracetamol after 15 hours under heating 220 degrees Celsius. Therefore, we found drastic condition is required, and sometimes high expensive catalyst is required, such as platinum nanowire, and it's not recyclable. Therefore, in our work, we started with baronitrophenol, and we used silver halide reduced graphene oxide as a photonanocatalyst. And we started to make enhancement to the for increasing the catalytic activity of this nanocomposite, and we succeeded to make this enhancement, and we obtained the paracetamol in very high yield, in 93% yield. After 8 hours by using silver bromide reduced graphene oxide. This work was published in Applied Organometallic Chemistry journal. Another research article, we started to make visible light triggered synthesis of bioactive tetrazole derivatives via click chemistry. We know about click chemistry, which is the formation of triazole or tetrazole ring. Therefore, we started to make this synthesis of the tetrazole ring by using photocatalysis technology. We made these analogs, 6 analogs, and then we started to make the anti-cancer activity of these analogs. We found that the tetrazole number 3, which bearing furan ring, it have high IC50 in for treatment A549 or lung, or this is the code of lung cancer cell lines. And also another tetrazole, it can be good treatment for breast cancer. Therefore, from I turn back to the previous slide. The tetrazole could be produced using visible light. And then it can be valuable product to make anti-cancer agents. This work was published in Journal of Organometallic Chemistry. Another work, we made microwave driven synthesis of propagelamine using platinum titanium dioxide nanocomposite. These 2 pharmaceutical products are known for treatment of Parkinson's and Alzheimer. These two known propagelamine. In our work, we made the synthesis of propagelamine using platinum titanium dioxide or silver titanium dioxide, and we found that platinum titanium dioxide was the best catalyst for this synthesis or for this protocol. We found that after 19 minutes, we obtained 82% yield therefore, it was a very good yield. And then we made optimizing condition to improve this yield and to improve and to see the effect of catalyst loading. Then after adding 0.25 mole percent of platinum titanium dioxide, we obtained 98% yield. So we succeeded to obtain valuable products such as propylamine by using recyclable platinum titanium dioxide in very excellent yield, in high yield. And this work was published in 2019 in chemical papers. Another recent article, which is ultrasound-assisted synthesis of tetrazoles derived from alpha amino acid side ester using zinc oxide nanoparticles. We know about the important value of amino acids, and it can be included in many pharmaceutical products. Therefore, we try to introduce tetrazole ring by using the amino acid as starting material. We used in this reaction zinc oxide nanoparticles to be as a photocatalyst, and we used ultrasound as irradiation in this protocol. And under optimized condition, we used 5% only of zinc oxide nanoparticle, and the solvent was a mixture between acetonitrile and water, and we obtained high yield of tetrazole analogs after 30 minutes only. And this work also was published in Applied Organometallic Chemistry Journal in 2020. Finally, I would like to conclude my presentation. The heterogeneous nanophotocatalysis in organic synthesis could be regarded as an economically and eco-friendly and green approach. It was observed that the photocatalytic reaction can be influenced by the different parameters. Heterogeneous photonanocatalyst in organic reaction is not limited only in the functional group transformation, such as oxidation or reduction. I mean, we can make tetrazole ring, we can make trizoles or another heteroaromatic ring, not only oxidation or reduction. The future perspective, the heterogeneous non-photocatalysis can be used for the activation of unreactive chemical bonds under mild reaction condition, formation of sp3 carbon-carbon. This means in the future, the scientist can develop the photocatalysis by making the reactions which is difficult to be performed under normal condition. It can be employed in the stereoselective synthesis, such as the last paper I presented in the synthesis of the tetrazole conjugated to amino acid. The possibility to find new synthetic approach in organic chemistry for the synthesis of valuable products. Finally, I would like to make acknowledgement for my research team, Professor Dr. Hussam al-Nazir, Dr. Yasser Atiyah from Cairo University, Gamal al-Ghannam, Cairo University, Isam Abdel Salam, Dr. Akabir Tariq from Zagazig University, Shayma Madani from Cairo University, and international contributors, Associate Professor Eric Solom, Carlos Vizgui, and my students, master degree and PhD degree student. Here is a picture was taken in 2019 with my master degree student and PhD degree student with Professor Hussam al-Nazir, Yasser Atiyah, and me. Thanks for listening. Thank you.
شكرا دكتور ياسر. ودلوقتي مع المحاضرة الأولى للقسم الامتداد الدكتورة شيماء موافي تحت عنوان Synthesis and Biological Evaluation of Antimicrobial Tetracyclic Cyclic Peptide. اتفضل.
Assalamu alaikum. My name is Dr. Shayma Mwefi. I'm a researcher at the National Research Center, Peptide Chemistry Department. I would like to thank you for giving me this opportunity to give this short talk. My talk is entitled Synthesis and Biological Evaluation of Antimicrobial Cell-Penetrating Cyclic Peptides. So I'll be talking about antibiotics and the importance of the antibiotics in the area of post-COVID-19 pandemic. So the antibiotics have been used effectively to treat bacterial infection. When there is no resistance and there is infection that occurs, when the drug treatment is used, the bacteria die. But when there is drug-resistant bacteria, as you can see in the red bottom line, red bottom figure, the bacteria multiply, there is resistance to the drug, and they continue to spread, and the person remains sick. What could be the cause for this resistance? So there are two major causes: genetic mutation, where basically the bacteria will do some genetic mutation, and which will make the bacterium drug resistant, and they will continue to multiply, and the drug will not be effective. The other reason for drug resistance is gene transfer. Gene transfer facilitates the spread of drug resistance, where basically bacteria that have drug-resistant DNA make a copy of the genes and to the other bacteria, and in the presence of the drug, only the drug-resistant bacteria will survive. So as you can see here from this schedule, the developing resistance is an emerging problem, and it continues to be a critical problem. Because as you can see from the yellow color, which is the date when the antibiotic is introduced, and the red color with the date, the timing where the antibiotic resistance is identified, you can see is that with time, the time frame between the introduction of the antibiotic and the resistance is getting very tight. So you always need to get new drugs to overcome the resistance that has occurred with the previous used drug. So COVID-19, and I'm quoting here from the website of the WHO, has poignantly reminded us that we need to build more resilient health system that include access to effective antibiotics to better tackle future outbreaks. Okay, and there is novel antibiotics are needed urgently now and more than ever, because with the pandemic, you need more effective antibiotics, and you need drug pipeline to come up with new antibiotics to promote and build the health system, so that patients that suffer from COVID-19 don't suffer also from secondary bacterial infection as a resulting of the COVID-19 pandemic. So For the purpose of this talk, I'm just focusing on antimicrobial cyclopeptide, and my talk will be divided into two different items. The first one is the cyclopeptides, if they could be synthesized within the bacteria cells, this work that I have previously done, and the new work that I'll be working on are cell-penetrating amphiphilic cyclopeptides. Both of them could be considered as the approach of our lab for the antimicrobial cyclopeptides. Concerning the cyclopeptides synthesized within the bacteria cells. So this is a rule of thumb for every new antibiotic that you want to develop. You always need to develop inhibitors that target a pathway that hasn't been targeted before, so that you don't come up with an antibiotic that has resistance as soon as it has been developed. So it will be a waste of money and waste of effort. You need to find always a new target. This target has to be involved in the virulence of the bacteria. It has to be conserved so that you can affect as many bacteria as you can. And it should be known and well studied so that it will be easy to develop an inhibitor easily for this target. When I was working on this project, we worked on a pathway called sigma E pathway. I'm just talking here, like, I want to, from my talk, to give like general rules in order to give ideas and share ideas with the audience about how to develop inhibitors targeting any future pathway if you need to come up with new antibiotics effectively and in a short period of time. So here, for example, the envelope stress response that are involved in gram-negative bacterial pathogenesis, with the sigma E pathway that I'm talking about is responsible for this envelope stress response, where you can see here that this sigma factor here is responsible for maintaining the cell envelope integrity. And whenever you target this sigma E pathway, you're gonna alter the composition of the outer membrane protein of the cell membrane, and this is gonna affect the life of the bacteria and affect the cell envelope integrity. I'm also showing how we worked out on this. So here, this is a different, how can I say this? This is a different idea than starting to do like synthetic libraries. We started the other way around, where we know the pathway that we want to target, and we develop inhibitors for this particular target. And here we developed something called reporter strain. And the idea behind this is to get a positive readout when you have inhibition of this pathway. Without going into details, which might be more related to molecular biology field, here the idea is that when I inhibit this pathway, I should see under the microscope YFP production, which will give me bright cells under the microscope. And this is great because I know that I have inhibited the pathway I wanted when I see bright cells under the microscope. And if I don't see any cells under the microscope that are bright, it means that I haven't found the proper inhibitor for my pathway. And we use cyclopeptides here as potential inhibitors because always cyclopeptides have considerable benefits compared to any types of other inhibitors. They have enhanced binding affinity, they have enhanced metabolic stability, and one of the major advantages in this technique that have been used is that there is an ability currently to biologically synthesize them within the bacteria cells so that we know they are produced within the bacteria, and based on that we can target and see whether the pathway that I'm focusing on has been inhibited or not. So how could we synthesize them within the bacteria cells? We clone them on a plasmid. So inside the bacteria, they will have different plasmids, and one of the plasmids will carry the expression of the bacteria, of the cyclic peptide. So as you can see here, also without going through details, here we use a technology called intane-based technology for peptides, which basically allow the cyclization within the bacterial cells. And this cyclization is a spontaneous reaction where we get rid of the intane N and C terminal, and the cyclization will happen, and we'll have small cyclic peptides cyclized within the bacteria and expressed. And so if they are really inhibiting the pathway, you will see bright cells under the microscope. So this plasmid, or this library, or cyclopeptide library that is within the bacteria, is called Cyclops, and it has been used for maybe like a couple of years right now. So it's based on a spontaneous reaction. It's expressed in vivo, and it encodes 3.2 to 10 to the power 6 different cyclic peptides. Every single peptide cell that is going to be bright is going to express a different cyclic peptide. So here, this library encodes 8 cyclic peptide composed of 8 amino acids, where we have the serine, the glycine, and the tryptophan are fixed, and then you have 5 different random amino acids, which are depending on the library that you expressed randomly from the cyclic peptide library. Very quickly, what we have done, since we're looking for bright cells, we sort the brightest cells using a technique called FACS. So as you can see here, this is my positive control, the one with the dashed line. And then this is my screening strain containing the peptide, and this is my screening strain without the peptide. And if I'm looking for the brightest cells, I'm looking here at the arrow with the red arrow so that I get the highest percentage of the brightest cells that are very close to the positive control. When I set up this in my sorting process, I collect the brightest cells, which are only 0.01% of total cells, and I rescreen them looking for bright cells under the microscope. And this is what happened. I'm showing you examples with 3 different plasmids encoding 3 different peptides. This is my positive control. You see the positive cells. And here with the plasmid expressing my peptide, I also see bright cells, meaning that the pathway that I'm looking for has been inhibited. So I know that the sigma E pathway, that is specifically the one I want to target, is already inhibited. And this is on the upper side is the negative control, which is only the strain without any peptide expression. So after that, we have to double check that this peptide really inhibit the growth of the bacteria. And this is what you can see here from this graph, where basically the growth of the E. coli has been looking at the optical density. You can see that the bacteria stop growing and they finally die. Also looking is using different stains with the propidium iodide stain here. You can see here with the red color here, it means that these cells are dead. So also is expressing my peptide. I know that the cells are getting killed and that the peptide is toxic to the cells. This is the sequence of one of the peptides that I was able to generate from the cyclopeptide library. And see, as you can see here, it's a cyclopeptide. It has serine, glycine, tryptophan. As we said, these are from the library. They are stable, they are fixed. And then you have 5 different other amino acids. And this is the combination of the peptide that has been shown to be active and to produce bright cells under the microscope. So this is one type of, or one way of how to get antimicrobial cyclic peptide without having to go through synthetic library. But if you want to go through synthetic library, the other part I'm going to talk about shortly is cell-penetrating amphipathic cyclic peptides. And also the cell-penetrating peptides is going to be the focus of the talk of my colleague, Dr. Shaimaa Mahdi, after me. So I won't talk a lot about this, but just to give you an idea about what we can do also as cell-penetrating peptides used for antimicrobial resistance. So what are cell-penetrating peptides? Very briefly, cell-penetrating peptides, they are small peptides of less than 30 amino acids. They are usually cationic, positive charge, so that they can be binding to the plasma, the membrane of the peptide, and they have the ability to enter the cells. They are not cell specific, and they also act as when I say that they're cell penetrating, they can also be used as vehicle for delivery of other peptides or other compounds that cannot be introduced or they cannot enter the cell properly, whether it's another antibiotic or another also drugs used as anti-cancer or for drug delivery. These are very good and delivery vehicle called cell penetrating peptides. So they target the negatively charged bacterial membrane. They enhance the delivery of cell impermeable peptides or other molecules. And one example of this is using cyclized polyarginine peptide, because arginine is positively charged. So when you have cyclized peptides, polyarginine that has more than one arginine, these are one of the classes of the cell penetrating peptides. So this is also a short diagram showing how it works. Basically, when you have an optimal balance of positive charge and hydrophobicity within the cyclopeptide, so you have hydrophilic and hydrophobic, you can better interact with the cell membrane, and this will allow better drug delivery of any hydrophobic molecule or cell impermeable molecule. And this is the work that has been done in the parent lab, where I had the opportunity to spend 9 months as a postdoc there, and I've done some interesting work. I'm showing you how they have been working on these peptides. So they found an antibacterial activity for amphiphilic peptides against multi-drug resistant pathogens. This peptide they called R4W4 because it's formed of 4 arginine and 4 tryptophan, and it's a cyclopeptide. They found that it has a very good MIC against MRSA, which we know is the methicillin resistance Staph aureus, that is very community acquired bacteria that we need to find antibiotics against them, against this particular species. So it had an MIC of 2.67 microgram per milliliter. It also showed activity against Pseudomonas aeruginosa and E. coli. And the most interesting thing is that they used it in combination with tetracycline, which is a known antibiotic. And guess what? When they did that, they found a better MIC when they use a combination between 2 to 8 folds against MIC and between against E. coli, and between 4 and 8 times better when it's used against MRSA. So the combination gives better MIC between this peptide and tetracycline. This is the structure. Again, when they use arginine, we know it's effective in terms of internalization. They have done a lot of works, and they found that the optimum ratio is to have 4 arginine and 4 tryptophan. The number of arginine is crucial. They said that between 7 and 15 residue will present an optimal uptake for the peptide. Considering the tryptophan, this is the hydrophobic residue that helps in the translocation across the membrane. These aromatic residues have favorable free energy of insertion, and they are also involved in membrane destabilization process. So this is the structure of R4W4. So I'm also giving you an example of what are the ideas concerning cyclopeptides to be used for antimicrobial resistance. Diverse application of this category of cyclopeptides, as you can see here, this is R4W4 or R5W5. They're showing both types because I've been working on both types of cyclopeptides. Look at the uses of the application of this cyclopeptide. It can be used as surfactant, nuclear targeting, molecular transporting, doxorubicin prodrug. So basically they bind, I'm gonna show you one figure for this, where they use the doxorubicin, which is a known antibiotic, and they use it as a prodrug by combining it with this cyclic peptide. They performed a lot of in vivo study. They use it as oligonucleotide and sRNA transporter, negatively charged phosphatide transporter. They even also use it with gold nanoparticle for drug delivery systems. It has different various applications, and some of them are useful for antimicrobial resistance, and others are useful for cancer therapy, for drug delivery in general, for any drug delivery that you want to target to a particular cell within the nucleus or within the DNA. So they have very many uses that can be used for. This is the figure I'm showing you. So basically, this is one example of the R4W4. They removed one of the arginine and replaced it with lysine. And then they added a linker so that they can bind the linker here with the lysine and the R4, which won't be R4, it will be R3, then lysine, and then you have the 4 tryptophan haven't been changed, and they bound it to doxorubicin, and this has been shown. They did all the work, and they have shown that doxorubicin penetration has been optimized using FACS sorting and other types of techniques to prove that the doxorubicin entry and penetration to the cell has been optimized when it's combined with the cell-penetrating cyclic peptide. One part of the work of my contribution when I was there is we were able also to synthesize, if you want to go even bigger, we were even able to synthesize multivalent tricyclic peptide, which is based from 3 different cyclic peptides And the good thing about it is that it's also linked between them with amide linkage, which is very natural and very easy to be degraded within the cells, and you don't have to introduce different or toxic catalyst or anything. It's going to be very safe, and this was shown with all the study that was done. And this is one type of tricyclic peptide that has shown to have modest antibacterial activity, but was very promising as molecular transporter for siRNA and anti-HIV drugs. So basically, it can also be used as a transporter for antiviral drugs, anti-cancer drugs, and other compounds. And this is based only on the use of cyclopeptides as a transporter. So what I wanted to show you here is that the many uses that you can use for the cyclopeptide in various fields, including the antibiotic resistance. So finally, I want to go back and make a general conclusion about the invaluable role of antibiotics in pandemic and beyond. Since the theme is about COVID-19, we need to keep reminded that we need to be prepared for any further public health crisis, whether COVID-19 or any future outbreaks, and antibiotics are needed to protect currently the COVID-19 patient who have weakened immune system. They might be on ventilator, they are risk of infection for secondary bacterial infection. And also antibiotics, even if we don't have this pandemic, they are crucial for patients fighting cancer, receiving dialysis, undergoing surgery, and other medical treatments and procedures. You cannot, you have to face the idea of antibiotic resistance, and you have to come up with new antibiotics. And I hope from this talk that I've shown you that cyclopeptides are very promising as candidates for antimicrobial resistance. And thank you.
شكرا دكتورة شيماء موافي على هذه المحاضرة. ودلوقتي المحاضرة الثانية لقسم الببتيدات بعنوان سيل بنتريتنج بيبتايد فروم سيري تو ابليكيشن للدكتورة شيماء مهدي. اتفضلي يا دكتورة.
مش عارفة ليه بس أي لا تمام.
السلام عليكم. My lecture today about cell-penetrating peptides from origin to application. It's an overview or general lecture about cell-penetrating peptide. So, first of all, cell membrane regulates exchange of materials from outside to inside the cells, and cell membrane is selectively permeable with no general mechanism for their uptake. And the process of introducing a molecule, a biomolecule, to the cell as a drug or any biomolecule can always be a major upscale or challenge at both research and industrial level. And successful systemic administration of any drug always include a series of steps, each of which has its own constraints. So the question is how to get a drug into cells through through the cellular membrane. Simply, the answer is cell-penetrating peptides, which is a method or approach that important breakthrough in cell biology, and the search in this field has gained much attention in recent years. There are many novel therapeutic approaches for drug delivery existing already, but with many limitations. These approaches, like gene and protein therapy, have slow or limitation in its potential due to the cell impermeable nature of peptides and oligonucleotides. So the existing methods for delivery of macromolecules, such as viral vectors or membrane perturbation techniques, can result in high toxicity, immunogenicity, and low delivery yield. The approach for delivery of macromolecule into target cells with high efficiency and efficacy is urgently needed. And cell-penetrating peptides, simply a technology that has that can overcome the membrane impermeability to afford the efficient delivery of bioactive cargoes. Cell-penetrating peptides Cell-penetrating peptides, also known as protein transduction domains, which are relatively short, up to 30 amino acid residue. They are water-soluble, cationic, and/or amphipathic peptides, which make them promising vectors for therapeutic delivery. But why cell-penetrating peptides? They penetrate the plasma membrane at low concentration without using any receptor and without causing any significant membrane damage. Also, they efficiently deliver biologically active cargoes such as drugs into cells, and they have low cytotoxicity compared to other cytoplasmic delivery systems such as liposomes and polymers. So they have potential application in the diseases diagnosis and therapy, including cancer, inflammation, central nervous system disorder, and even diabetes. The history of cell-penetrating peptides. Actually, the first discovered cell-penetrating peptide was in 1988 when they studied the HIV type 1 virus, human immunodeficiency virus type 1. They found that it properly enters the cells and can deliver even many molecules. So they discover there's about 13 amino acid region exists in the residue between 48 and 60 region residues in whole protein. So it became the first cell-penetrating peptide in 1988. The second one was in 1994, which is penetratine. It's a 16 amino acid from the third helix of Antimicrobial homodomain. And the following cell-penetrating peptides was in 1997, which is the first synthesized cell-penetrating peptide, which MBG, and TET, a truncated arginine. Following that, in 1998, first chimeric cell-based peptide was introduced by Langel group, which is part of it's like a mixture of neuropeptide galanine linked to masteroperon and transportane peptide were formed. Since then, a hundred of natural peptides, synthetic or natural therapeutic peptides, were continued to grow, and this table represents many synthetic or natural discovered cell-penetrating peptide up to now, and classifications. Actually, there's many route of classification could be applied for cell-penetrating peptide, and based on many factors and properties. First of them, based on origin, like we see here, based on origin to protein derived, which represent 42% of the total number of discovered peptide. It's a naturally occurring protein, lactate and penetratin, as I mentioned before. Also, chimeric cell-penetrating peptide. It's we can say between natural and synthetic, because it's could contain parts of natural or synthetic peptides, can come together or link together with covalently or non-covalently bonded, which represents only 3%. And the synthetic cell-penetrating peptide, as we see, 55% of the total existed peptide, cell-penetrating peptide, which is fully synthetic. The most famous one is polyarginine. Also, another way to classification according to conformational structure of the peptide, which is linear or cyclic. Of course, many or the most cell-penetrating peptides that discovered or synthesized is linear, as we see about 95% of the total existed peptides. Another one, which is the most important one, route of classification, is based on physical chemical properties of the cell-penetrating peptides, which include cationic, which mainly or based on the positive charge amino acids inside the sequence, arginine, lysine, or histidine. Amphipathic cell-penetrating peptide, which have lipophilic and hydrophilic moieties in the same cell-penetrating peptide, and the hydrophobic. The most of cell-penetrating peptides are cationic or amphipathic, actually. Another route or type of classification depend on or based on the type of linkage between cell-penetrating peptide or the peptide and the its cargo, and the type of its application inside the cell. So let's move to another point. Uptake mechanism. The question is how cells can uptake the cell-penetrating peptide itself or with cargo. First, we can speak about the complex or linkage between cell-penetrating peptide and cargo. The linkage mainly non-covalently or covalently. Non-covalently, cargo's charge, it's a charge-dependent bond, and covalently, which is a bond, dipeptide, disulfide, sulfonyl, and many others. According to this type of linkage, be the penetration. So the uptake mechanism actually is not well known until now, and they found that the same cell-penetrating peptide has many routes or ways of internalization, even with the same cells. But it can depend on many other factors like types of cells, temperature of incubation time, and also the complex, the interaction between peptide and its cargo. Many other factors affect actually this process, but mainly the mechanism method has two ways: direct translocation or endocytosis, or the normal endocytosis. Direct penetration contain inverted micelles or barrel stave model or carpet-like model, and endocytosis involved clathrin-mediated endocytosis, caveolin-mediated endocytosis, clathrin, caveolin, independent endocytosis. Actually, it depends, as I mentioned, on many factors. Prediction. Up to now, many in silico cell-penetrating peptide prediction approaches were established to facilitate the identification of potential cell-penetrating peptides. The publicly accessible cell-penetrating peptide site, it's a database is a store of about 1,850 validated cell-penetrating peptide sequences, and a valuable source of information regarding model systems and the range of cargoes delivered by individual sequences. One valid approach towards cell-penetrating peptide identification employ QSAR, or quantitative structure activity relationship results, to a computer algorithm highlight the cell-penetrating peptide sequences with the primary sequences of whole proteins, and especially within sequences enable protein-protein interactions, which often contain one or more cationic helix. The websites Cell PBD and Skip CBB Bread are alternative and freely accessible platform that employ computational method to predict and modify cell-penetrating peptide sequences. Here we can see an example of prediction of a peptide sequence, VG21. A represent the structure of the peptide. B represent the results of the, as I mentioned, computerized or mathematical score, SVM. It's a mathematical score, gives every part or sequence of in the peptide a score. This is score maximum to 5, but indicates the most important or most effective, potentially effective in internalization of the cells, and give it a colored orange, green, and blue for every sequence. And C, D, E, F represent many properties of this peptide, like 4D structure and physiochemical properties, and charging, hydrogen bonding, and so on. So everything is computerized before we can decide what cell-penetrating peptide we can organa to synthesize. We can already mathematically or theoretically know its potential. Therapeutic application of cell-penetrating peptides. For peptides and protein delivery. Delivery of a biological active protein like beta-galactosidase linked to TAT peptide. So TAT, beta-galactosidase across the blood membrane it's an example for peptide and protein delivery for cell-penetrating peptide. This conjugate TAT with galactosidase across the blood-brain barrier was improved after intraperitoneal administration. Also, for drug delivery, cell-penetrating peptides conjugated to small molecules like drugs in case of cancer, shiferdine, and imaging agent to increase their intracellular delivery and to overcome drug resistance. For nanocarriers, recently cell-mediated peptides, including TAT peptide, were successfully used to deliver different nanoparticles, pharmaceutical carriers, like nanoparticles, liposomes, and micelles into the cells. For imaging agents, also, CBP-mediated delivery was used to label cells with QDs as fluorescent probes. Also, TAT-conjugated peptides were used for an effective labeling of the cells, like immune cells, for in vivo magnetic resonance imaging goals or for bioimaging purpose. As antimicrobial activity, CPP-based antioxidant containing the aromatic and cationic residue indicated to be very effective for mitochondrial delivery. For vaccination, the cell-mediated peptides were suggested as a promising agent for vaccine delivery, especially DNA, protein, peptide-based vaccines. CPPs were often fused with antigen to achieve efficient cell membrane translocation, enhancing antigen uptake, processing, and presentation by antigen-presenting cells. For cancer therapy, recently cell-penetrating peptides were used to deliver chemotherapeutic drugs like Taxol, cyclosporine A, and methotrexate, and pro-apoptotic proteins into resistant cells and improve their activity. Also, multifunctional CPPs were developed with their physiochemical properties to overcome the heterogeneity and resistance of the tumor's tissue to tumor-related biomarkers. Actually, for a recent application of cell-mediated peptide in my PhD study on a protein called c-KIT. It's a stem cell factor receptor on mast cell, in attempt to know or control the degranulation of mast cells as a reaction to any drug. We choose as receptor already exists in mast cells and apply QSAR prediction algorithm, a type of prediction algorithm to this mathematically or computerized program. We apply this whole protein that contain about 900 amino acids. After this, we result in many sequences with high index for penetration. This followed by senses, microsenses of these chosen peptides, and of course, biologically screening of the synthesized peptide for secretion, for internalization of the cells, for cytotoxicity, for the tumor cells and normal cells. The result was very good about and compared with our positive target, positive control TAT. This chart indicates the quantitative assessment of cellular internalization of the synthesized peptide, and I think most of the synthesized peptide were relatively good in internalization of mast cells compared with TAT protein or peptide. Some disadvantages for cell-penetrating peptide are their short half-life in blood and non-specific CBP-mediated delivery to normal tissue. This problem could be improved by coupling CBP to other carriers like liposomes and the use of endogenous and exogenous stimuli like specific enzymes or pH value, or even mild heat, respectively, to increasing cell-penetrating peptide-mediated therapeutic efficacy. Up to now, few cell-penetrating peptide-linked drugs have entered the clinic for both topical and systemic administration. The first compound that entered phase 2 clinical trial was a cyclosporine A polyarginine conjugate for the topical treatment of psoriasis in 2000. To conclude, the efficiency of cell-penetrating peptide to penetrate the cell membrane and deliver different cargoes to specific intracellular sites is a suitable approach for delivery of chemotherapeutic. Although many preclinical studies shows a promising result through the cell-penetrating peptide-mediated delivery of therapeutic molecules in treating cancer and other disease, no CPP-based therapeutic approaches has approved by FDA yet. Thank you.
بس المشكلة إن هي حضرتك كانت على الفلاشة مش في الفولدر، فهو ده اللي
حضرتك اطلعي طيب. بس للناس في حاجه هنا مهنجه. لا لسه كمان كده لازم تفصيل. بس حاجه انا عايز اقفل البرامج دي مش اللي هي ايه تمام.
في الاول باوربوينت هو الباوربوينت اللي عمل القصه
بس هو دي اللي كنت عايز اجيبها.
بس خلاص المحاضرة بتاعتي ده في الفولدر اللي عندك. مين؟
ريت.
فيديو.
اشتغل.
اسفين على الوقت اللي حصل. دلوقتي مع محاضرة قسم الكيمياء العضوية الفلزية والعضوية شبه الفلزية، حيث يعد هذا القسم أحد أهم أقسام شعبة بحوث الصناعات الكيميائية الذي أنشئ بقرار من السيد الدكتور رئيس المركز القومي للبحوث عام 2005. أما الأهداف والاستراتيجية للقسم فهي تشييد المركبات العضوية الفسفورية الفلزية وتصنيع المركبات الفلزية ذات النشاط البيولوجي والصناعي. كما يهدف القسم إلى استخدام المحفزات الفلزية وشبه الفلزية والإنزيمات في تشييد المركبات العضوية والتفاعلات الفراغية. كما يهدف القسم إلى تحضير المواد الوسيطة للعقاقير ومتشابهات العقاقير والأدوية. ويعمل القسم على تحقيق هذه الأهداف ويطورها، وذلك حتى يتماشى مع رؤية مصر 2030. والآن مع المحاضرة الأولى للقسم للدكتور فريد محمد سرور، أستاذ باحث مساعد بالقسم، تحت عنوان Recent Progress in the Organometallic Chemistry of the Cyclooctatetraenide Anion. اتفضل يا دكتور.
بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله. هتكلم النهارده إن شاء الله عن Recent Progress in the Organometallic Chemistry of the Cyclooctatetraenide Anion. جميل. المحاضرة هتتكون من introduction cyclooctatetraenide anion ligand مع الالكالاين ميتال، الالكالاين ايرث ميتال، ترانزيشن ميتال، لانساميد اند اكتينايد ميتالز. وبتتكون المحاضرة الأولا هنتكلم عن الانترودكشن. الواحد 3 5 7 سايكلوكتاتيترين اللي هو مختصر بكود، بيبقى شكله في كأورجانيك كومباوند واخد تيوب شيب، او احيانا بيسموه بنانا او تشير شيب. في الحقيقه المركب ده بيكون نون اروماتيك نون لينير، لكن لما بيحصل له ريدكشن ب 2 الكترون بيتحول للسايكلوكتاتيترانايد، وده ليجند مختلف تماما في الاروماتيستي. طبعا ده بيبقى اروماتيك، بيبقى بلانر، والكيمياء بتاعته بتختلف تماما عن الكود العادي قبل الريدكشن. فزي ما احنا شايفين ان لما بيحصل ريدكشن ل 1 3 5 7 سايكلوكتاتيترين ل 2 الكترونز بيتحول للسايكلوكتاتيترانايد انايون او داي انايون. في الحقيقه اول واحد عمل التفاعل ده كان كاتس حضر الليجند ده عن طريق الريدكشن للكود باستخدام الميتال البوتاسيوم ميتال في الدي ام اي في التتراهيدروفيورين والدي ام اي والامونيا والتيميدا اللي تتراميثايل ايثيلين دايامين. بعد كده في طرق جديده برضو حديثه تم تحضير فيها الكود داي انايون باستخدام اللي هي 1,5 سايكلو اوكتاداين. وبعد كده برضه بيديني اللي هو البوتاسيوم كود. البوتاسيوم الكود او الكود داي انايون ليه ديريفيتيف مختلفه زي ما احنا شايفين في الحلقه في ترايميثايل سيلايل ممكن بيبقى فيه 3 جروبس او ممكن 2 بالاضافه طبعا احيانا بيبقى فيه فينايل. ففي ديريفيتيفز للسايكلو اوكتاتيترين ليجند ممكن برضه تستخدم في الاورجانوميتاليك كيمستري ويبقى لها ابلكيشن كويس جدا. هنتكلم عن البارت الاول اللي هي السايكلو اوكتاتيترانيل دايناميك كيمستري اوف الكالاي ميتالز. زي ما قلت من شويه كانت سنه 1960 حضر اول كود بالداي بوتاسيوم سولت بتاع الكود عن طريق الميتال البوتاسيوم ميتال مع السايكلو اوكتاتيترين. عملها في الامونيا طبعا اول مره تمت في الامونيا في -30. وبعد كده بدات الطرق تعمل موديفيكيشن بيحضروها في التي جي اف او في الدي ام اي او في البنتان احيانا. بعد كده جه فريتز عمل فيرست كريستال لاين للكود تي جي اف ده كان سنه 1960. The first crystal structure of lithium code has been reported in the group of Professor Edelman in Germany. في الحقيقة كان هو أول واحد دكتور بروفيسور إيدلمان في ألمانيا عمل أول فيرست كريستال ستراكشر للليثيوم كود. وزي ما احنا شايفين في الكريستال ستراكشر إن الكود ساندويتش ما بين 2 ليثيوم ايونز كل واحدة كوردينيتد مع 3 من الكربونز بتاعة الكود. زي ما احنا شايفين الايتا 3 للكومباوند ودي كان حاجة unusual في الليثيوم كود كيمستري ان هو قدر يفصل الكريستال ستراكشر بتاع الليثيوم. هو لحد دلوقتي مش غير الليثيوم كريستال ستراكشر ده اللي اتفصل ككريستال ستراكشر للليثيوم كود. الانترستنج هنا ان مش كل الكربونز بتاعه الكود كوردينيتد للليثيوم ايونز. في 3 من كل 3 مختلفين عن بعض زي ما احنا شايفين ان ايتا 3 الاين لايك فورم ان هي الكود ساندويتش ما بين الليثيوم 3 هنا و3 الناحيه الثانيه وده مش مشهور كتير. لورنس ايتال ريبورتد ده حديثا 2018 كان ريبورتد لعمل بوتاسيوم كود كيمستري ومنشوره في الاورجانوميتاليك كيمستري دي مجله كبيره قويه في التخصص. كان عمل استادي جميله ان هو حضر بوتاسيوم كود كيمستري مختلفه باختلاف الكونديشن. عايز اقول ان الكيمياء بتاعه الكود بتعتمد باعتماد كبير بتتاثر باي حاجه سواء كان السولفنت او الكونديشن اللي تم فيها التفاعل او الكاتاليست اللي تم او حتى نوع الميتال اللي بيتم. الكيمياء بتاعته بتعتمد كتير جدا على الحاجات دي. يعني اقل حاجه ممكن يغير لي حاجات كتيره في البرودكت اللي يطلع معايا ودي ميزه طبعا في الكود ان انا بقدر اعمل كنترول شويه على البرودكت اللي هيطلع معايا. زي ما احنا شايفين السايكلو اوكتاتيترين ديريفاتيف لما بيحصل له ريدكشن بالبوتاسيوم هيدرايد في الدي ام اي بيديني بوتاسيوم كود كومباوندز زي ما احنا شايفين 3 و4 و5 و6. في حاله الدي ام اي مع الكي اتش بيديني لما يكون الديريفاتيفز على السيليكون الار هنا بتختلف طبعا. لما تكون ريسايل مثلا بيديني 2 كومباوندز اللي هو 3 و4 مختلفين عن لما يكون الديريفاتيفز اللي على السيليكون اللي هي الار هنا مثلا لما تكون مثلا تيرشر بيوتايل او فينايل. لما نشوف مثلا في حاله الميثايل ديريفاتيف هنلاقي ان حصل شيفتنج للترايميثايل سيلايل من 1 و4 ترايميثايل سايكلوكتاتيترين ل1 و3 وده طبعا في الكيمياء ما بيبقاش بسهوله. وادانا بالبرودكت الثاني حصل ريموفنج لمجموعه من السيليكون ترايميثايل. في حاله وجود الترايفينايل او تيرشر بيوتايل في كسبستيوشن على السيليكون بيديني 1-3 مركب واحد بس اللي هو 5 أو 6. في حاله الترايفينال بيديني 6، وفي حاله التيرشروبيتايل ميثايل بيديني 5. طبعا هو الكيمياء بتاعت الكوت لازم تتفصل بالسنجل كريستال، ما نقدرش الان ام ار وحده ما يقدرش يقول لي بالظبط البرودكت. لذلك هو لازم عشان اقدر اقول ان التفاعل مشي في الطريق ده وده دايما الكريستال زي ما احنا شايفين
ورق النايل
المركبات تم فصلها كسنجل كريستال اكس راي ستراكشر. لحد دلوقتي اصله في السنجل كريستال، لكن في كومبيتيشنال ستاديز تمت على الكيمياء بتاعت الصوديوم كوت، وقالت ان هو مش هيختلف كتير عن الليثيوم او البوتاسيوم، ان هو هيبقى برضه الكوت ساندوتش ما بين تو ميترز اوف صوديوم، برغم ان هيبقى في بيحصل احيانا تنافس شويه ما بين الصوديومز، بس المتوقع ان هيكون في السنجل كريستال او في السوليد ستيت هيكون نفس الاستراكشر بتاع زي ما احنا شايفين بنفس الاستراكشر بتاع البوتاسيوم او الليثيوم. ودي كانت كومبيتيشنال ستاديز تمت على الكيمياء بتاعت الصوديوم كوت. ندخل على النقطه الثانيه اللي هي سايكلو اوكتاتيترانايل داينايون كيمستري في الكالاي ايرث ميتال. الالكالاي ايرث ميتال اللي هي المجموعه الثانيه في البريودك تيبل، برغم ان الايونيك ريديوس بتاعها قريب جدا من الايونيك ريديوس بتاع اللانسانايد بلس 2، لكن الكيمياء بتاعتها تختلف تماما عن اللانسانايد بلس 2. زي ما احنا شايفين مثلا الكالسيوم قريب جدا الايونيك ريديوس بتاعه من اليوتيربيوم بلس 2، والسترونسيوم قريب من اليوروبيوم بلس 2 برضه. بس الكيمياء بتاعت الكود في اللانسانايد مختلفه لما تتم، حتى سوريتيكال لما تم دراستها لقوها بتختلف عن اللانسانايد ايون. لحد دلوقتي كيمياء قليله جدا تمت على المجموعه الثانيه اللي هي الالكالاين ايرث ميتال باستخدام الكود كسراوندنج ليجند. لذلك انا في 2017 خدت بوست دكتور وسافرت فرنسا، اشتغلت مع البروفيسور ميشيل إتيان واقترحت عليه نعمل الكيمياء. انا كان عندي خبره طبعا في الكيمياء دي من الدكتوراه في المانيا، فاقترحت عليه نعمل الكيمياء بتاعه الكالسيوم والسترونسيوم باستخدام الكود كسراوندنج ليجند، وهو بصراحه رحب وما قال مفيش مشكله نجرب الكيمياء الجديده دي. وفعلا بدانا زي ما حضراتكم شايفين في السكيما اللي على اليمين. لو خدنا تفاعل الميتال داي ايودايد مع البوتاسيوم امايد مع الكود سولت او الكود بوتاسيوم سولت، يعني المولر ريشيو هنا بيختلف، بيقدر يعمل لي كنترول للبرودكت. يعني مثلا لما افعل مولر ريشيو في رقم A مثلا بمولر ريشيو 2:1 هنلاقي ان الكود ساندويتش ما بين 2 calcium atoms or 2 strontium atoms and even every metal atom bonded with amide and TGF. طبعا زي ما قلت المولر ريشيو، طبعا في الحاله دي البوتاسيوم ايودايد لما بيحصل بريسبيتيشن، هي مشكله الكيمياء المجموعه الثانيه ان بيحصل فيها الايكوليبريا ان التفاعل بيفضل رايح جاي من الشمال لليمين، يعني البرودكت يدفع تديني الرياكتنت والرياكتنت يدفع تديني برودكت. لذلك الكيمياء بتاعتها مش مشهوره حتى في العالم، مجموعات قليله اللي شغاله في الكيمياء بتاعت الجروب 2 الكالسيوم والسترونسيوم والباريوم. لكن في حاله التفاعل ده كان الجيد ان البوتاسيوم ايودايد زي ما احنا شايفين بيترسب، وده بيعمل بوشنج للتفاعل من الشمال لليمين، وبالتالي بيخلي التفاعل بيمشي يعني سموثلي عشان يكون لي الساندويتش كود كومبلكس زي ما احنا شفنا اللي هم 7 و8. في حين ان لو نفس الرياكتنت تم تفاعلها بمولر ريشيو مختلف 1:1 زي ما احنا شايفين في الطريق في باس 3، هنلاقي ان الكود ساندويتش ما بين 2 هيتيرو ايونز اللي هم الكالسيوم والبوتاسيوم. الكالسيوم الكود اخذ مول من الكالسيوم ايودايد ومول من البوتاسيوم كود، والكالسيوم طبعا بالامايد. هو نفس التفاعل 9 ده فتم فصله طبعا بالسولفيتد والانسولفيتد كومبلكس، هو نفسه لو فعلته مع الكالسيوم ايودايد كوان مول والبوتاسيوم امايد كوان مول هيديني المركب اللي هو 7 و8 تاني. وبالتالي دي كانت طريقه جميله ان انا اعمل كنترول شويه على البرودكت اللي بيطلع معايا، وان انا اقدر احضر حتى الانترميديت لو اعتبرنا ان رقم 9 ده الانترميديت. ومن هنا كانت الكيمياء سهله جدا ان احنا نقدر نتحكم في البرودكت في فصل الانترميديت. هو الانترميديت 9 نفسه لو فعلناه مع سترونسوم ايودايد، سترونسوم امايد، بيديني اللي هي المالتي ديكا ساندويتشز رقم 10. زي ما احنا شايفين ان الكوت ليجند ساندويتشز ما بين 2 سترونسوم اتومز، وكل سترونسوم اتومز بايندد بامايد وامايد بوتاسيوم. طبعا امايد بوتاسيوم ده بيبقى كوردينيتد، ما هوش من ضمن البرودكت، ولكن في حاله السوليد ستيت بيحصل كوردينيشن للبوتاسيوم مع الامايد والامايد مع السترونسوم. هو رقم 10 ممكن يتحضر بطريقه ثانيه، بطريقه مباشره جدا زي ما احنا شايفين في الباسوي تحت، السترونسوم ايودايد مع بوتاسيوم امايد مع البوتاسيوم كوت في وجود تي اتش اف ل 2 ل 1 2 ل 4 ل 1 مولر ريشيو، بيديني دايركتلي رقم 10. زي ما احنا شايفين ان كل مركب بيحصل عندنا كنترول في البرودكت شويه من خلال الرياكتنس المولر ريشيو اللي انا بقدر ابدا بيه في البدايه، اقدر اتحكم في البرودكت. ودي كيمياء جميله، ودي كانت اول مره حد يشتغل براكتيكال. انا بتكلم براكتيكالي، كان اول حد يشتغل على الكيمياء بتاعه الكالسيوم والسترونسيوم في الكود كيمستري. طبعا كل المركبات دي تم فصلها سنجل كريستال اكس راي زي ما احنا شايفين كلهم، عشان اقدر اقول اذا كان البرودكت ده طلع معايا صح ولا لا. زي ما قلت من الاول ان الكيمياء بتاعت الكود لازم يكون في سنجل كريستال اكس راي ستراكشر، لان زي ما قلت ان الكيمياء بتاعته بتعتمد اعتماد كبير على السولفنت، على الرياكتنت، على الكونديشن اللي بتتم، على البرودكت اللي بيطلع معايا. وبالتالي اقل حاجه بتحصل تغيير في التفاعل بيديني برودكت مختلف. حاولنا نجرب الكاتاليتيك اكتيفيتي بتاعت الكومبلكس 9، الكالسيوم كود بوتاسيوم اللي هو الهيتيرو اتومز كود كومبلكسز، بتكوين الكربون نيتروجين بوند عن طريق تفاعل الفينايل اسيتيلين مع الكربوديمايد، ده ايزوبروبايل كربوديمايد او سايكلوهكسال كربوديمايد لتكوين الكربون نيتروجين بوند اللي هو الجوانيدين يعني. وفصلنا الانترميديت بتاعه زي ما احنا شفنا لما نفعل مولر ريشيو 1 ل1 فينال اسيتيلين مع الكاتاليست، بيدي قدرنا نفصل رقم 12 كومبلكس 12 اللي هو بيبقى فيه الكالسيوم ساندويتشد ما بين 2 كوت، وايتش كوت ده مالتي ديكر، وده اول مالتي ديكر يتم فصله في كيمياء الالكالاين ايرث ميتال. زي ما احنا شايفين ان each coat ligand sandwiched ما بين 1 calcium 1 coat and each calcium atom sandwiched ما بين 2 coat. ودي كانت اول زي ما قلت اول كومباوند تم فصله في كيمياء الجروب 2 للكود. بعد كده بعدها بسنتين كان في هيل في مارسي، في جامعه مارسي، حضر مركب برضه كوت بالكالسيوم عن طريق تفاعل اللي هو الدايكيتيمينات ده اراي البوتاسيوم هيدرايد مع 1357 سايكلو اللي هو سايكلو اوكتاتيترين في ماينس 25 ديجري. زي ما احنا شايفين بنلاحظ ان بيديني الساندويتش كومبلكسز اللي هو فيه الكود ساندويتش ما بين 2 كالسيوم ايونز. زي ما قلت ان الكيمياء بتاعت الالكالاين ايرث ميتال بتاعت الكود قليله جدا، ويمكن من 5 سنين او 6 سنين بدات الشغل فيها بعد ما احنا بدانا يعني في تولوز. كان قبل كده كل الشغل بيبقى ثيوريتيكالي، بيتم دراسه معمول ابحاث كثيره ثيوريتيكالي على النقطه دي، لكن براكتيكالي كان شغل قليل جدا اتعمل. وزي ما قلت لان الكيمياء بتاعت الالكالاين ايرث ميتال كمان مش مجموعات كتير على مستوى العالم شغاله فيها. لو كل دوله في اوروبا هتلاقيها مجموعتين 3 أو 4 بالكتير شغالين على الكيمياء بتاعت الكالسيوم والسترونسيوم والباريوم. في ابحاث كتيره ثيوريتيكالي درست البايندينج ما بين الميتالز والكود كومبيوتيشنالي يعني، وتوقعت البايندينج هيتم ازاي. طبعا الباريوم طبعا باعتبار ان هو اكبر ايونز في الجروب، معتقد ان هو ايتا 8 دايما هيبقى البايندينج بتاعه ايتا 8 مع الكود. الكالسيوم نفس القصه والسترونسيوم، وبالتالي اغلب كل المجموعات هتبقى دايما الكربونز اتومز بتاعه الكود دايما كوردينيتد مع الميتال دايركتلي از ايتا 8 كوردينيشن مود. الترانزيشن ميتالز طبعا المعروف ان الترانزيشن ميتال ليها مميزات خاصه في البيريودك تيبل. كل الكوردينيشن مود تم ملاحظته في الكيمياء بتاعه الكود، بدايه من الايتا 2 لحد الايتا 8. يعني قصدي ان كل الكربونز بتاعه الكود تحصل كوردينيشن ما عدا ايتا 7. يعني ما تمش لحد دلوقتي there is no papers have been reported this coordination mode. يعني ايتا 7 الوحيده، لكن باقي الكوردينيشن مود بدايه من ايتا 2 لحد ايتا 8 ان كل الكربون بدايه من 2 كربون لحد كل الكربون بتاعت الكود بيحصل كوردينيشن مع الميتال، وريبورتد في ابحاث كتيره زي ما هنشوف دلوقتي. طبعا اول تفاعل كان كلوك في انجلترا، كان ريبورتد للسكانديوم اللي هو unusual code complexes زي ما احنا شايفين رقم 14. في 1989 كلوك ريبورتد الكومباوند ده عن طريق التفاعل بتاع السكانديوم ترايكلورايد HF مع كوت-ليثيوم. زي ما احنا شايفين ان السكانديوم ساندويتش ما بين 2 كود، وايتش سكانديوم اتومز بريدجد ب 2 كلورين اتومز. في تي اتش اف طبعا بريدجد ما بين التو سكانديوم. في 2002 رابي كان عم ريبورتد لفيرست 6 ري ستراكشر للسكانديوم اللي هو الانيوجوال برضه. زي ما احنا شايفين ان السكانديوم طبعا سكانديوم بلس 3 السكانديوم اتومز في كومباوند رقم 15 السكانديوم ايونز ساندويتشد ما بين 2 كود، ولكن في واحد فيهم ايتا 8 الكل ال 8 كربون بتاع الكود كورنيتد مع الايونز، في حين الليجند الثاني الكود 4 بس، والاربعه الثانيين كورنيتد مع الليثيوم. وده كان انيوجوال لان زي ما نشوف في السنجل كريستال اللي على اليمين هنلاقي ان الاربعه كربونز بتوع الكود الكورنيتد للليثيوم مش هم نفسهم الكورنيتد للليثيوم. ودي فليكسبيليتي بتبقى موجوده في الكود دايما، ان هو يعني زي ما احنا بنقول بيميل شويه ناحيه البوزيتيف شارج بتاعت الايونز، ففي بيديله فليكسبيليتي اكتر ان هو يكون ستيبل كومبلكسز مع باختلاف السايز ايونز بتاع السايز ايونز بتاع الميتالز. ومن هنا ده كان unusual complexes reported في 2002. السكانديوم بايميتاليك داينيوكلر بايميتاليك هاف ساندويتش كود زي ما احنا شايفينه في 2017 تم فصله السنجل كريستال. طبعا ده دايما بيستخدم سواء كان السكانديوم او اللانسانايد او الاكتينايد دايما بيستخدم كستارت ماتيريال في تحضير مركبات اورجانوميتاليك كتير. فده برغم ان الكيمياء بتاعته يعني المركب ده قديمه شويه، ولكن اول سكانديوم كود بالشكل ده تم فصله في 2017. وده يدل ان الكيمياء دي لسه جديده جدا وناس قليله اللي شغاله فيها، وانترستنج ان احنا نقدر نفكر فيها هنا في مصر. حديثا براونشفايج عمل استادي جميله حاول يشوف تاثير السبستيوشن على الكود ليجند في الكوردينيشن مود بتاعه مع الميتالز، وعمل دراسه مع السكانديوم. هنا زي ما احنا شايفين في 3 كومبلكسز 16 17 و18. لما بيفعل السكانديوم ترايكلورايد مع 3 ليجند مختلفين وبيشوف السبستيوشن اللي هو البورون امايد دايميثايل زي ما احنا شايفين كده، لما يكون السبستيوشن موجود في السي بي اللي هو سايكلوبنتاداينايل ما بيأثرش حتى لو كان في السي بي بس او في الكود بس زي ما احنا، او مش موجود يعني في كومبلكس 16 هنلاقي ان مفيش سبستيوشن تم في الكود او في السي بي. الكوردينيشن مود زي ما هو بتاع الكود، حاله السبستيوشن بقى في السي بي برضه ما حصلش تغيير، وفي حاله وجوده في رقم 17 وفي حاله رقم 18 لما يكون السبستيوشن في الكود برضه ما حصلش. وبالتالي فان السبستيوشن على الكود ليجند ما بيلعبش دور كبير في الكوردينيشن مود بتاع السايكلوبتاترانايل ليجند في السراوندنج فيلد بتاع الميتال ايونز. برضه التيتانيوم هنا زي ما احنا شايفين بيتفاعل مباشره التيتانيوم امايد مع الكود وبيكون لي الهاف ساندويتش تيتانيوم امايد كومبلكسز 20 و21 بنفس الطريقه برضه كومباوند 22 في الامايد برضه في وجود الديريفاتيف، بس تحت لما معاهم بنستخدم مولر ريشيو مختلف بيديني الهاف ساندويتش كومبلكسز 23. كل المركبات دي تم فصلها بالسنجل كريستال اكس راي ستراكشر. كان راوش في المانيا وتلميذ دكتور بروفيسور ايدلمان، كان عمل استادي جميله برضه لما يعمل بنت 3 نوفل زيركونيوم كومبلكسز، اما يديني البنت برضه زي ما احنا شايفين انه ما اثرش برضه على الكوردينيشن مود بتاع الكود بالنسبه للزيركونيوم، وكل 8 كربونز وداني الايتا 8 في وجود يعني مثلا في كومبلكسز 22 و 23 رغم ان في زيركونيوم بوندد بكلورين الكوردينيشن مود زي ما هو للكود، لما بستبدل بالفينايل بيديني الكوردينيشن مود زي ما هو. وطبعا كل الثلاثه مركب 22 و23 و24 سيبريتد سنجل كريستال اكس راي ستراكشر. هنشوف اول حاله كان الايتا 3 كان بالتنتالم ميتالز، ان حصل كوردينيشن مود ايتا 3 زي ما احنا شايفين في الاكس راي ستراكشر، ان التتاليوم بايندد بثلاثه كربونز بس للكود ليجند ودي كانت اول كوردينيشن مود ايتا 3 بيتم هنا، حصل ريبورتد وده كان في 1998 تقريبا، ودي كانت اول حاله للترانزيشن ميتال في الايتا 3 للكود. نفس القصه في الرونيوم ميتالز، احنا شفنا هنا لما بيتم تكوين الرونيوم كود كومبلكسز مش كل الكربونز بتحصل كوردينيشن مع الرونيوم، في 2 كربونز ما بيدخلوش ككوردينيشن مع الميتال، ودي برضو اول حاله للايتا 6 كوردينيشن مود للكود في ترانزيشن ميتال زي ما احنا شايفين في كومبلكسز 34. طبعا الترانزيشن ميتال فيها ميزه مش موجوده في كل الميتالز الموجوده في البريودك تيبل، ان بتكون الميتال بوند، والميتال بوند دي تديها ميزه مختلفه شويه عن باقي الميتالز في البريودك تيبل، ان هي بتقدر يعني يبقى في فلكسبيليتي اكتر في البايندنج مع الكود. زي ما احنا شايفين ده الفيجر ده بيلخص شويه الكوردينيشن مود بتاع مثلا الحديد، ممكن الايرون ممكن يكون كوردينيشن مود ايتا 4 مع الكود، الكوبالت ممكن يكون ايتا 2 مع الكود، في حاله التيتانيوم طبعا ممكن يتكون تيتانيوم بونز، وبالتالي تم فصل مركبات فيها الايتا 5. في مركب مثلا اللي فيه الحديد ممكن يحصل كوردينيشن مود للايرون مع الكود ب2 مود مختلف لنفس الكود ليجند ايتا 5 وايتا 3 زي ما احنا شايفين مع الايرون، وبالتالي الميزه دي مش موجوده في اللانسانايد، مش موجوده في الاكتينايد، مش موجوده في باقي الميتالز في البريودك تيبل، ودي كانت مميزه جدا في الترانزيشن ميتال. طبعا نلاحظ ان الكود نقدر نصنفه كويك ليجند لانه بسهوله ممكن يحصل فيه فليكسبيليتي، يعني الدلتا جاب بتاعته ما بين الاوربيتالز مع الترانزيشن ميتال قليله، وبالتالي الموفينج بتاع الالكترونز بيبقى سهل ان هو يحصل، وبالتالي بيتصنف كويك ليجند، وبالتالي هيبقى فيه فلكسبيليتي اكتر في الترانزيشن ميتال. في ثيوريتيكال ستاديز كتيره تمت في البايندنج ما بين الترانزيشن ميتال والكود ليجند، زي ما احنا شايفين بناء على الميتال بوند بيبقى دايما في اختلاف على حسب الكوردينيشن سفير حوالين الميتال بوند. النقطه الاخيره السايكلوكتاتريال داينايون كيمستري في اللانسانايد والاكتينايد كيمستري. طبعا اللانسانايد والاكتينايد نتيجه السايز بتاع الايونز بتاعتها ان هي كبيره بيبقى دايما محبذ ان هو الكيمياء بتاعته دايما مع الكود، لان بيحصل ايتا 8 دايما، او يعني مش دايما، اغلب المركبات مع اللانسانايد بتتم كايتا 8 كوردينيشن مود. في 2007 كان لورنس عمل حضر اللي هو الكود توبليوم كومبلكسز، وشاف ان اختلاف السولفنت بيدي ما بين 44 بي و44 اي، زي ما احنا شايفين ان السولفنت بيلعب دور مهم وبيكون بيعمل ريفورميوليشن للكومبلكسز بطرق مختلفه. فهو حضره في الدي ام اي بطريقه مختلفه، وحضره في بنسب مختلفه، وقدر يفصل ال 44 A وال 44 B برغم ان هو نفس الكومبلكسز، ولكن باختلاف السولفنت قدر يديني كومبلكسز مختلفه. زي ما قلنا قبل كده ان الساماريوم، ان الباي ميتاليك كومبلكسز بيدي نقدر نستخدمه مركب 41 مثلا، ويستخدم كستارت ماتيريال في كيمياء كتيره جدا في الكود كيمستري، وبالتالي بيبقى تحضيره مهم جدا، وفصل السنجل كريستال بتاعته مهمه جدا. برضه مركب 45 زي ما احنا شايفين برضه لورنس فصلوا باستخدام المالتي ديكر كومبلكس. زي ما احنا شايفين ان البروزيديميوم بيتفاعل بروزيديميوم ترايكلورايد في التي اتش اف مع التولين مع الليثيوم كوت ليجند، بيديني الهيوج اورجانيك ان اورجانيك كومبلكسز اللي احنا شايفينه. وده برضه دي نتيجه الفلكسبيليتي بتاعت الكوت ليجند اللي تقدر تديني فورم مختلف في كل حاله من الايونز. برضه تفاعل الليثيوم اللانسانايد كوت ليثيوم مع لما يحصل مع الكوبالت دايكلورايد بيديني المالتي ديكر. زي ما احنا شايفين النوديميوم ممكن يكون لي ساندويتش كومبلكسز، واليوتيربيوم ممكن يديني المالتي ديكر ساندويتش كومبلكسز. في 2014 كنت خدت دكتوراه من المانيا، وكان الشغل بتاعنا حضرنا بعض اللانسانايد كود كيمستري. زي ما احنا شايفين ان احنا لما بناخد ستارت ماتيريال لانسانايد كلورايد كود كستارت ماتيريال مع الاميدينيت بيديني الهاف ساندويتش كومبلكسز اللي بتبقى فيه الاميدينيت بريدجد ما بين التو لانسانايد اند ليثيوم ايونز. نفس الطريقه تحت لما بناخد السيريوم اميدينيت كومبلكسز مع الكود بنلاحظ ان الكود ساندويتش ما بين تو سيريوم ايونز اللي هم 57 و 58 زي ما احنا شايفين، يديني الهاف ساندويتش كومبلكسز. نفس الطريقه برضو بالبريزيدوميوم كود مع الاميدينيت بيديني البريزيدوميوم هاف ساندويتش كود كومبلكسز اللي بتكون فيه الاميدينيت ساندويتش ما بين تو بريزيدوميوم ايونز. لكن في حاله الهولميوم زي ما احنا ملاحظين ان الهولميوم بيبقى السايز بتاعه اصغر شويه، فما كونليش الباي ميتاليك كومبلكسز في المركب 63 و64 نتيجه السايز بتاع الهولميوم. ده كان الاستراكشر كان انترستنج جدا ان احنا نفصل الاكس راي ستراكشر بتاعه اللي هو رقم 65 اللي بيكون فيه النوديميوم الكود ساندويتش ما بين تو نوديميوم ايونز، وايتش نوديميوم كورنيتد بتو كلورينز بريدجد مع النوديميوم اللي جنبها. وده كان هيوج جدا و12 في الاورجانوميتاليك. في 2019 كان بيتر خينا فصل المركب ده اللي هو بتاع الميتال كود اللي بيكون الاتريوم واللانسانيوم. ودي كانت حاله نادره ان بيبقى فيه الكود كوردينيتد بايتا 3 زي ما احنا شايفين في مركب 66، ودي بتبقى قليله قوي في اللانسانايد. فهو قدر يفصل المالتي ديكر ساندويتش ده، وبيكون فيه الكود كوردينيتد بثلاثه كربونز مع الاتريوم او اللانسانايد. السنه اللي فاتت كان الدكتور روسكي فصل مركب اللي هو بوتاسيوم ب C9H9، وده مفصول وده نشره في النيتشر ان هو فعل البوتاسيوم صلب بتاع C9H9 مع اللانسانايد كود، وقدر يعمل ساندويتش كومبلكسز من اللانسانايد كود و C9H9، ودي قدر يفصلها في النيتشر كوميونيكيشن السنه اللي فاتت دي، وده كان ستراكشر كويس جدا. اخر نقطه سايكلوكتاترانين مع الاكتينايد. طبعا الاكتينايد اللي هي السوريوم والبلوتونيوم واليورانيوم. المركبات دي بتكون بتكون كومبلكسز كويسه جدا مع الكود. زي ما احنا شايفين في السوريوم بلس 3 كان مركب 71 تم فصل هيوج كومبلكسز للسوريوم مع الكود، وبنلاحظ ان هنا each كود ليجند ساندويتش ما بين السوريوم والبوتاسيوم في مركب هيوج، وده كان اتفصل لاول مره. التفاعل الثاني برضه في استخدام البلوتونيوم. البلوتونيوم اتفصل برضو بلوتينيوم 4، اتفصل الميتالوسينز المركب 72 زي ما احنا شايفين بطريق مباشر جدا، وده كان اول مره يتفصل البلوتونيوم 4 بطريق مباشر من تفاعل الليجنز الليثيوم كوت مع بلوتينيوم ترايكلورايد. النقطه الثانيه، النقطه اللي قبل الاخيره، البلوتونيوم بلس 3 بيتم تحضيره برضو مع الكوت، وحصل له اوكسيديشن وتكوين البلوتونيوم 4. طبعا دي في جامعه كاليفورنيا تم تحضير البلوتونيوم بلوتونيوم كوت بلس 3، وبعدين بيحصل له اوكسيديشن لتكوين البلوتونيوم 4. ودي كان اول مره برضو في جامعه كاليفورنيا. طبعا كل الشغل ده بيبقى دايما بالذات في الاكتينايت بيبقى ليه لايسنس مختلفه لان هي راديو اكتيف كومباوند. الهدف من الكلام ده ان احنا نقول ان الكيمياء دي جديده ومهمه، ان احنا ممكن نفكر ان احنا نشتغلها في مصر طبعا، لان زي ما احنا شايفين ممكن تفاعل واحد يتنشر كبحث في احسن المجلات في التخصص. فكنا عايزين نعمل سبوت على النقطه دي، وشكرا جزيلا.
شكرا دكتور فريد على هذه المحاضرة القيمة. دلوقتي مع المحاضرة الأخيرة للقسم تحت عنوان سيليكا سلفوريك أسيد إيثيلين جلايكول ايكو فريندلي كاتاليست فور 1 بوت سينسايز اوف نيو بنزا اميدازو بريميدينز. اتفضلي يا دكتورة نجوى.
السلام عليكم، معاكم الدكتورة نجوى مراد، قسم كيمياء عضوية فلزية وشبه فلزية. إن شاء الله محاضرتنا النهاردة هتكون under title Silica Sulfuric Acid Ethylene Glycol an efficient eco-friendly catalyst for 1-pot synthesis of new benzimidazopyrimidines. In view of recent trends in organic synthesis, investigation of new and hazardous chemical reactions have become a priority in organic synthesis and are the most important protocols in organic synthesis as well as medicinal chemistry. One of the currently used areas of organometallic chemistry involves the development of new environmentally benign catalytic systems. The use of metal reagents has received considerable importance in organic synthesis due to their ease of handling, greater selectivity, enhanced reaction rate, and recoverability of catalysts. The use of eco-friendly chemical reagents as catalysts in organic synthesis reduces materials, energy, time, waste, hazards. The first part about multi-component condensation reaction of 2-amino-benzimidazole, aldehydes, and dicarbonyl compounds. This part was published in Organic Preparation and Procedure International Journal. Number 1, reaction of 2-amino-benzimidazole and aldehyde with acetylacetone. Optimization of the reaction position was achieved by taking the reaction of 2-amino-benzimidazole, 4-fluorobenzaldehyde, and acetylacetone as model reactants under different conditions. The reaction afforded the corresponding 1,4,4-fluorophenyl to methyl 1,4-dihydroethanone 1A in body lights. From the optimization condition, when carried out the reaction in ethylene glycol without catalyst, it was produced the desired product in moderate yield, 50%. Then using different catalysts, namely silica-supported polyphosphoric acid, perchloric acid adsorbed on silica gel, and silica sulfuric acid. It was found that the best catalyst in terms of yield and reaction time, silica sulfuric acid produced 93% in 15 minutes. Moreover, when studying the efficacy of the catalyst ratio, we found that 0.11 produced the best ratio, 0.15 73, 0.025 85%. Then replacing ethylene glycol with different solvent, water, methanol, ethanol, chloroform, we found that ethylene glycol produced the best yield, 93%. After that, chloroform, 75%, water and ethanol, nearly the same year, 74% and 58%. The scope and limitation of the three-component reaction under optimized condition were explored using a variety of aromatic and heterocyclic aldehydes. When two amino benzimidazole and acetylacetone were left to react with different aldehydes, the reaction afforded the corresponding benzoimidazopyrimidine derivatives 1A to G. From the reaction with different aldehydes, we found that 4-fluorobenzaldehyde reduced the base yield 93%. After that, 3-chlorobenzaldehyde 83%, benzaldehyde and 2-cyphenaldehyde reduced the same yield 80%, 2-naphthaldehyde 75%, but 5-methylphenaldehyde reduced the lowest yield 60%. Proton NMR spectrum of 1G as a representative example exhibited two singlet signal at 2.2 and 2.4 for 2-methyl proton, and singlet signal at 6.8 for CH proton of the pyrimidine ring and pro signal for NH group. And carbon 13 assignment 2 signal at 20 and 30 for 2-methyl carbon, and singlet signal at 56 for CH carbon of the pyrimidine ring. Number 2, reaction of 2-amino benzimidazole and aldehyde with benzoyl acetone. When benzoyl acetone was left to react with different aldehydes and 2-amino benzimidazole in the presence of silica sulfuric acid and cylinder alcohol, the reaction afforded the corresponding benzoimidazopyrimidine derivatives 2A to G in lights from 58% to 97% depending on the nature of the aldehyde. Like the reaction with acetylacetone, 4-fluorobenzaldehyde reduced the 97% the base yield. After that, benzaldehyde 92%. 2-naphthaldehyde and 3-bromobenzaldehyde reduced the same yield 75%. 3-chlorobenzaldehyde and 2-cyphenaldehyde reduced nearly the same yield 72 and 73%. Proton NMR of 2A exhibited singlet signal at 1.8 methyl proton and singlet signal for 6.6 for CH proton of the pyrimidine ring and pro signal for NH group. And the carbon 13 assignment signal at 90 for methyl carbon and signal at 56 for CH carbon of the pyrimidine ring. Number 3, reaction of 2-amino benzimidazole and aldehyde with 1,3-diphenyl propane 1,3-diol. When 2-amino benzimidazole and 1,3-diphenyl propane 1,3-diol were left to react with different aldehydes in the presence of silica sulfuric acid and ethylene glycol, the obtained product were identified as dihydropyrimidine benzimidazole derivative 3 rather than the expected product dihydropyrimidine benzimidazole 2-phenyl methanone derivatives. In this reaction, molecule of benzoic acid toast and form compound 3. From the reaction with different aldehydes, we found that benzaldehyde and 5-methylphenaldehydes reduce the base yield and the same yield 97%. After that, 2-naphthaldehyde 95%, 4-fluorobenzaldehyde reduced 94%, and 2-cyphenaldehyde 76%. Proton NMR of 3A exhibited 2 doublet signal at 5.2 and 6.3 for 2CH proton and pro signal for NH group. And carbon 13 assignment signal at 5.56 for CH proton of the 4CH carbon of the pyrimidine ring. Part 2, multi-component condensation reaction of 2-amino benzimidazole, aldehydes, and active nitride. This part was published in Synthetic Communication Journal under this title, Reaction of 2-amino benzimidazole and aldehyde with malonitrile. In order to optimize the reaction condition, 2-amino benzimidazole, benzaldehyde, and malonitrile were taken as model reactants under different conditions. The reaction afforded 2-amino 4-phenyl 1,4-dihydrobenzoimidazopyrimidine 3-carbonitrile 4A. From the optimization condition, the reaction without catalyst in ethylene glycol produced the lowest yield 50%. Silica sulfuric acid reduced 97% the best yield. After that, perchloric acid 90% and polyphosphoric acid 84%. Moreover, open studying the efficacy of the catalyst ratio, 0.11 produce the best yield. After that, 0.025 87 and 0.15 90%, 0.05 84%. The replacing ethylene glycol with different solvents. We found that ethylene glycol reduced the best yield 97% in 5 minutes. After that, ethanol 84%, methanol and dimethylformamide reduced the same yield 77%, and chloroform and water produce nearly the same yield 74% and 72%. After that, 2-amino-benzimidazole and malonitrile were left to react with different aldehydes. The reaction afforded the corresponding 2-amino for aryl 1,4-dihydrobenzoimidazopyrimidine 3-carbonitrile derivative 4A to F. From the reaction with different aldehydes, it was found that benzaldehyde, 2,5-dimethoxybenzaldehyde and 2-cyphenaldehyde produce the same and best yield 97%. After that, 4-fluorobenzaldehyde 87%, 4-hydroxybenzaldehyde and 3-methoxybenzaldehyde produce the same yield 83%. Proton NMR of 4E as a representative example exhibited two single signal at 3.6 and 3.64 for 2-methoxyproton, and single signal at 5.3 for CH proton of the pyrimidine ring, and broad signal for NH2 and NH at 6.8 and 8.2. And the IR spectrum of 4E exhibited strong absorption bands for cyano group. Reaction of 2-amino-benzimidazole aldehydes and ethyl cyanoacetate. The three-component condensation reaction of ethyl cyanoacetate with 2-amino-benzimidazole and different aldehydes leading to two products. The obtained products were confirmed as 1,2,3,4 tetrahydropyrimidine derivatives 5A2G and 1,2-dihydropyrimidine derivatives 6A2G rather than the expected product 7 and 8. The mechanism of this reaction. Silica sulfuric acid serve as lowest acidic catalyst for condensation the aldehyde and ethyl cyanoacetate, leading to the formation of araldine ethyl cyanoacetate 1. Since the amino group of 2-amino-benzimidazole reacted with the later intermediate 1 in the Michael way, forming intermediate 2, which is subjected to loose molecule of ethanol and cyclization, forming the desired product 5, which underwent dehydrogenation from compound 6. From the reaction with different aldehydes, we found that in all cases except 3-chlorobenzaldehyde, the majority product compound 5, while compound 6 was observed as a sole product in 3-chlorobenzaldehyde. The final reaction of 2-amino-benzimidazole aldehydes and cyanoacetamides. For the optimization condition, 2-amino-benzimidazole, 4-fluorobenzaldehyde, and cyanoacetamide were left to react under different conditions. The reaction afforded compound 10A rather than compound 9. The probable reason for this phenomenon, compound 9 easily react with another aldehyde and form compound 10A. From the optimization condition, it was observed that the reaction with ethylene glycol without catalyst produced moderate yields. Then using different catalysts, silica sulfuric acid produced the best yield 99% in 1 minute. After that, perchloric acid 93%, and after that, polyphosphoric acid 87%. Moreover, when studying the efficacy of the catalyst ratio, we found that the same 0.11 produced the best yield 99%. After that, 0.1593, 0.0259%, and 0.0587. When replacing ethylene glycol with water, methanol, ethanol, chloroform, and dimethylformamides, ethylene glycol produce the BCl 99 in 1 minute. After that, ethanol 87. Methanol and dimethylformamide produce the same yield 80%. Water and chloroform produce nearly the same yield 75 and 77%. After that, 2-aminobenzimidazole and cyanoacetamide were left to react with different aldehydes. The reaction afforded tetracyclic dihydropyrimidine derivatives 10A to E rather than the expected product tricyclic dihydropyrimidine derivatives 9, compound 9, easily react with another aldehyde and form compound 10. From the reaction with different aldehydes, 4 fluorobenzaldehydes and 3 methoxybenzaldehyde produce the same and BCl 99% in 1 minute. After that, 3 bromobenzaldehyde 89%, 2 naphthaldehyde 86%, and 3 chlorobenzaldehyde produce the 80%. They make cytotoxic activity of some benzoimidazopyrimidine derivatives. The present work aims to screen the synthesized compound for testing their potential to inhibit the viability of cells into human cancer cell lines, namely breast and hepatic carcinoma. The in vitro cytotoxic effect of the compound was tested against African green monkey kidney and 2 human tumor cell lines, HEBG2, and human breast carcinoma, MCV7. 1 compound 2E were reaction of 2-aminobenzimidazole with benzoyl acetone and 2 naphthaldehyde out of 38 gave remarkable cytotoxicity more than 75% effect and was able to inhibit the proliferation of the cancer cell, HEBG2, whereas the cytotoxicity of the other compounds range from moderate 75 to 40% to weak less than 40% effect. Thank you.
وبانتهاء هذه المحاضرة نختم فعاليات اليوم الثاني من الورك شوب تحت عنوان New Trends in Organic Sciences and Their Applications. بنحب نشكر كل السبيكرز اللي شرفونا النهارده وبرضو وحدة المؤتمرات على الجهد المبذول معانا، وبنفكركم لسه الورك شوب مستمرة يوم الثلاثاء ويوم الأربعاء تحت رعاية الأستاذ الدكتور محمد هاشم رئيس مركز القومي للبحوث والأستاذ الدكتور سيد التومي رئيس شعبة البحوث الصناعات الكيماوية. دمتم بخير وبالتوفيق.